Thank you very much, Dr. Suchek. I appreciate it. It's a real pleasure to be here. Pleasure to start this conference off. I hope for a really nice couple of days and some great discussion and hopefully start some collaborations. Let me tell you a little bit about myself. I work for the National Cancer Institute over in Maryland, so a quick jaunt down the road. I actually have been there for many years. I'm in a laboratory called Chemical Biology. It used to be medicinal chemistry, and things now have to be kind of biological, so we are now chemical biology. It turns out that we have been this name for the last 5 years. I just want to put, introduce some of the collaborators before I start. At the NCI Frederick, my current postdoc is Charlotte Chambre. Shabbat Biswas worked on some of the research that I'll tell you about at the end. Jack Bull is a technician in my lab. Scott Medina and Howard Young are both biologists that actually helped us with some of the biology. At the University of Buffalo, Kate Rittenhouse Olson, many of you might know. She's an immunologist who's helped us with some of the animal work that we did with some of our constructs that I'll show you, along with her student, Padmini Sabu. At the University of Maryland, we work with Susan Key, who's an infectious disease expert in another project that I probably will not tell you too much about today because I won't have time. And Alice McCarroll, who's a computational chemist who helps us with modeling of some of these glycopeptides that we work with. Okay. Since we do have maybe a little bit extra time, I do have a few extra slides, so hopefully we won't go too long, but if not, the organizer can yank me off. So most of you know that carbohydrates are very important in biology, and they function in many different ways. This is a really nice picture I like from a Japanese website that shows cell surface carbohydrates. There are things like proteoglycans, there's N-link carbohydrates, O-link carbohydrates, and glycolipids, and these have very different functions. And so proteoglycans might be involved in signaling, N-links in protein folding, glycolipids in microbial infection, and O-links in immune recognition. This is just a sampling of what their activities are, but we are actually interested in a particular type. And it turns out that they're very multifunctional, as I say here, but they're dramatically altered during tumor regenesis. So cancer has very, very different carbohydrates than normal cells, okay? And I'll try to tell you a little bit about what those differences are in the next couple of slides. We are particularly interested in O-link carbohydrates, and so I'm going to talk exclusively about those today. Okay, so in tumors, while N-links actually get bigger and more branched, and we can talk about why they do that later on, O-links get trimmer. And one of the things, a little picture from Essentials of Glycobiology, is that in the biosynthesis of O-link glycans, they start by adding one sugar at a time, right? And it turns out that if there's any kind of aberration in the enzymatic, in some of the enzymes that will transfer other sugars, they will halt at a particular place. So in other words, they might be only one sugar, like say the TN antigen or the TF antigen, is sometimes called a T antigen. Sometimes they get sialylated by the overexpression of sialyl transferases, and so they would be prematurely capped, okay, at places like sialyl TN or sialyl TF. And what that looks like, these are sometimes called tumor-associated carbohydrate antigens, because now that they're foreign carbohydrates now, so the immune system recognizes them as foreign. So it actually raises an immune response to them. Okay, and this is what we're very interested in our lab. So if you see a dramatic, graphical outline of, say, a cell surface protein, say like a mucin, okay, you'd see like lots of trees of carbohydrates that could be 10 to 12 residues. After neoplastic transformation, you might see these small sugars that are only 1 or 2. So now that does two things. It has an aberrant glycan that gets recognized by the immune system, and it does that because it exposes now the peptide backbone. That peptide backbone is not actually recognized over here when you have those big trees, because mucin proteins are proteins that are very, very large. They have maybe 1 million molecular weight, and they extend out from the cell surface, and these carbohydrates sometimes interdigitate. They protect the cell from outside inside. So now when they look like this on tumors, the physical properties, of course, of those cells are totally different, okay? So this TN antigen, as I mentioned, is simple. The alpha GalNAc, this is just lactosamine, right, in an alpha position, to either serine or threonine. The one we're interested in is called the Thomson-Friedenreich antigen, or TF. We could also get, again, sialylation at these positions to make other tumor antigens. The TF antigen is very interesting to us for several reasons. It's found in carcinomas, but it's rarely found in normal tissue. So you only really find it in tumors. It's a marker of aggression and metastasis, and I'll talk a little bit about that. And it's an aberrant glycan that activates a humoral immune response, like I just mentioned. Okay? At the same time, humans contain naturally occurring anti-TF antibodies. And those that have more anti-TF antibodies actually do better when they have tumors that express this particular sugar, okay? Because they have more antibodies to actually protect you from metastasis. TF antigen has also been employed in vaccine contracts, and there's several people that have worked in this area, okay? Actually, George Springer did this work back in the 70s and did an incredible amount of work. In 1980, published a paper that showed that if he made ghost red blood cells that actually exposed, that had presented the TF antigen, he injected these into patients that had breast cancer. It was very, very aggressive, as a matter of fact, and he got incredible responses. 5 years later, people were still living. So it's one of actually the best ones available. Several people have tried to make vaccines for TF, and none of them, some of them have reached phase 2, some early phase 3, but efficacy is not very good, and there's reasons for that we can discuss later. So there's nothing out there right now against the TF antigen. Antibodies to TF are also antimetastatic in vivo, and that's our collaborator has shown that. And carbohydrate mimics the TF actually inhibit metastasis in vivo. And that's another collaborator of ours that has worked in this area. So some models have been proposed which suggest that tumor cells that have TF antigen that express on, say, mutants such as MUC1, okay, will circulate in the vascular system of an individual during metastasis and actually interact with either endothelial-derived or circulating galectin-3. Galectin-3 is a lectin that binds TF very, very well. And when that interaction occurs, there's a repolarization on the cell surface where some of those mutants come together and they expose other proteins, especially integrins. Okay? Integrins are cell adhesion proteins that really slap things together very tightly. And so when a cell interacts with the endothelial cells that have integrins on them, it'll stay. It's able to form an embolus of tumor cells there and be able to extravasate and metastasize. It will not be able to metastasize if it doesn't hold itself there. Okay? And so this model has been shown in breast and prostate cancer, as well as pancreatic cancer by other workers. So Lu Gang Yu and Liverpool has shown this for prostate cancer, and actually Surinder Batra at Nebraska has shown that for pancreatic cancer. So what we set out to develop many years ago is a multivalent platform for TF. Now, why multivalent? I mean, most of you know that sugars, when they bind proteins in a monomeric way, it's very, very weak interaction. It's millimolar. Okay, so you need the Velcro effect. You need multivalency. You need lots of sugars on one kind of unit that binds to a protein, and you get much higher avidity, right? Affinity and avidity. And so what we decided to do is think about a platform where we can decorate them with these tumor-associated carbohydrate antigens or glycopeptides, okay, which was interesting to us. And in addition, possibly decorate them with things like T-cell epitopes or other molecular adjuvants that we could use to actually design possibly a vaccine construct out of this, okay? So we could either design something that will inhibit sugar-protein interactions in this way, or a new vaccine platform. So we wanted to use one platform for both, okay? And so our approach was to use gold nanoparticles. And so it was about 10 years ago or 11 years ago that it was just starting to show its face. Some carbohydrates were being presented on gold in synthetic ways, and it turns out that they've been very useful as glycobiology tools. They're very, very nice platforms because they're really, really easy to make. You can control the size and shape and the antigen and auxiliary molecule selection and ligand coding. They're actually non-immunogenic. So if you know anything about gold colloids, it turns out that they've been in humans for actually a long time. As a matter of fact, if you go to a health food store, you could actually buy gold colloids in a bottle. It's supposed to be an immune stimulant, as a matter of fact. So it actually it's supposed to have immune stimulating properties. So it's very interesting, the whole history behind this. But the synthesis of it is very simple. You can actually control the size by just changing the synthetic procedures. And again, they've been in humans, and we've put them in animals, and they're extremely non-toxic. So it turns out that use of tumor-associated glyco or glycopeptide antigens as immunogens sounds like a great idea to use with these particles. And so again, a dual purpose could be, they could either be anti-adhesion or they can be a vaccine. You can use them for either or, okay? There is actually one gold particle that's in clinical trials right now. It's in phase 2, and the NCI is sponsoring that trial. It's actually doing fairly well. It's a particle that has tumor necrosis factor alpha. And what it does is that tumor necrosis factor is extremely toxic to tumors, but if you inject that protein, you get dose-limiting toxicity, incredibly high. I mean, the therapeutic window is very bad. If you put it on a particle, it turns out that the toxicity goes way, way down, and so the efficacy is much better. So what about these constructs? What we made over the years are, we've made ones with just TF. We link them to a polyethylene glycol and a little fatty piece like close to the gold particle for actually packing. We've made them with that are attached just to the amino acid, like I showed you, because they're attached on proteins. They're attached to the serine and threonine. We put them on serine and threonine on the particle. And what we've done is we've also made what we call hybrid tumor-associated carbohydrate antigens, gold particles, where what we do is we put the TACA on there and we dope in a linker, okay, to actually reduce the density. And it turns out that has several different effects, which we can discuss later. I really can't talk about it now, but suffice it to say, these can be prepared in lots of different ratios. Okay. Many years ago, we also made quantum dots. If you've ever heard of quantum dots, very, very interesting imaging tools that are available commercially now. We actually made them with sugars on them, and we actually made ones that emitted in a near IR, but I won't talk about that today. So my first postdoc that I worked on this was Sergei Swarovsky. He is now in San Diego working for Zen Biosystems. He prepared these particles first. He did some studies where we showed that they bind beta-galactose binding lectins, they can agglutinate TF antigen binding specific antibodies and inhibit. We did an in vivo study where we got very, very nice inhibition of metastasis. But we only got that once. We weren't able to reproduce that. We're still trying to figure that out. We also looked at immune response when they were in the animals, and it turns out you get no immune response. If you just inject these particles, you get nothing. We're doing this without adjuvant, okay? We're just trying to see if this is going to work. It also does things like it turns on specific mitogenic signaling, like HERT1 and 2. And in an experiment we published last year, we can show that we can actually pull down galectin-3 and alpha-3 beta-1, which is associated with galectin-3 during the activation of endothelial cells in a system that mimics basically the metastasis. So using that particular particle, we're able to pull down galectin-3, alpha-3, and the beta-1, and much more than controls. And the control we used was just a particle that we coated with the linker. Okay, so just the linker, so just exposed OHs. So no sugar, but still a particle. And it was much better than control. It actually pulled down several proteins that are involved in focal adhesion, and this is also part of this whole particular process. So looking at these again, we also, again, as I mentioned, we have prepared these particles where we had them on the amino acid, because many people in the literature will say that the antigen is not just the sugar, the antigen is the sugar on the protein, right? So many people would make them on amino acids. What we want to do, I think the amino acid is not enough. You need to make the peptide also. So these have selective cytotoxicity of certain lymphoma cells, and I'm going to explain to you in a few slides later on. But what I wanted to tell you was that we wanted to extend this platform to multivalent glycopeptides, because our feeling was that the sugar needs the peptide, okay, in order to be immunogenic. That's the real immunogen, is the sugar on the peptide, because each one can influence the three-dimensional structure of the other, right? And so could we make multivalent glycopeptides and get immune responses? And so we used a particular mucin called MUC4, and I'll explain to you why we did that initially, but here's one of our constructs where we put sugars on different positions. We made linkers made out of polyethylene glycol. We were able to make these. This is actually NMR, believe it or not. You can take an NMR of the gold particles and actually get all of the fine structure, just a little bit broader because it's a much larger molecular weight. Our linkers here were actually fairly problematic, so we made new linkers, and this is the linker we use for everything right now, and it's actually very robust. And so we put, again, sugars at different positions on this, and we actually did vaccinations with the gold particles without any adjuvant. We also get no response. Just keep that in mind. So then what we did is with Ray Brenius, who came in about 4 or 5 years ago. Ray was a postdoc who came from a laboratory that actually worked a lot with gold particles, and we tried to design a new vaccine construct. And so what do we need? We need the appropriate platform, and gold was fine. We need the appropriate antigen, and that's our mucin-derived glycopeptide. We need the appropriate immune stimulant. So we used a molecule that we call a molecular adjuvant, and I'll tell you what that molecule is in a minute. And we need appropriate ligand density. So we actually need to do that doping experiment that I told you before. We need to dope in some of the linker, okay? And so what we did is we made these hybrid TF vaccine, we call vaccine constructs. And what do we use? So the TACA, of course, is TF, and the peptide is coming from a particular mucin, and the mucin we chose was MUC4, Okay, this is the 16 residue repeat unit of mucin 4. So mucins, the extracellular portion of mucins, many of them are basically lots of repeating units of between 16 and 24 or so amino acids. MUC4 in particular, this particular one has 16. And what is MUC4? So it's a mucin that's found on human tissue, but it's abnormally expressed in lots of cancers. And it's the only tumor-associated mucin that occurs on pancreatic adenocarcinoma. As a matter of fact, it's actually a biomarker for pancreatic tumors. So if you find aberrant MUC4, you'll find that if you have an adenocarcinoma of the pancreas. So in vivo models also show that overexpression promotes tumor progression and metastasis, so it's a very, very important one. And it turns out MUC1, those of you who are close to this field, MUC1 is the most important one, and people use MUC1 20 lots and lots of times. We decided to use this one because it was actually not explored at all. And so the initial synthesis used this particular peptide called C3D. So this is a peptide from the complement-derived protein C3, the D subunit. It's a 28 residue peptide that was mapped as the binding site, as a binding peptide for CD21. The CD21 on B cells is a receptor that, when it engages with this particular peptide, lowers the activation of the B cell to actually churn out antibody. So it's been used as a quote-unquote adjuvant for carbohydrate-based vaccines in the past. So we decided to put that on the particle too. So here's our design. We put our mucin 4 type glycopeptide where we have sugars at all different positions. We have our interstitial linker, which we dope in for density, and we have the C3D peptide, which is this particular peptide hooked to our linker, okay, in different ratios. And we get statistically significant immune responses to these particular vaccine constructs when we inject these into animals. This was a very, very large study, and I can tell you about the details later, but it was like 20 animals per group, and we had 4 different glycopeptides. We had a peptide itself, only the peptide, we had only the linker, and we had PBS. So we had lots and lots of different controls, and it turns out we get statistically significant responses, and sometimes IgM, sometimes more IgG, like for instance for this particular construct. But after we actually, at the same time, we actually implanted tumors in these animals to see if they would protect. And it turns out we didn't get very good data on that, but it turns out one of them actually rose to the top. This particular one called serine 5 glycosylated peptide, which is shown here. This one actually looked pretty good, and so we decided to follow up on this, and it turns out it's a fairly solid lead. So we get a survival advantage, with this particular construct versus control, even with very low levels of antibody. They get lower tumor weight and less severe metastasis, and treatment of mouse macrophages, which we've done in a collaboration with Howard Young, elicits selective production of TNF-alpha. So we've made several different particles with different types of things on them, and we actually show that with the 5 TF, actually we get a turn on gene expression of TNF-alpha. So it's an inflammatory response, and it turns out that just the peptide will do it, but at the 6th position it won't. But when you put the 5 back on, it recapitulates. So it's kind of an interesting whole study. This just shows that if we knock out MYD88, which is an adapter protein that actually turns on toll-like receptor synthesis, we actually knock down the expression of TNF-alpha. So it's going through toll-like receptors. We don't know which one, but that's a work in progress. So what we did was we took we took all of our constructs and put them on a glycan array. So Jeff Gildersleeve works in our lab, and so we collaborate a lot, so we put it on his array. And what we did was I went out many years ago, and when I knew about the 5TF, I sent out the sera to get to a company. I sent the construct to a company to get polyclonal sera made. When we got that made, we brought it back, put it on the array, and it turns out that sera only bound our peptides. So Jeff's array has about 300 or 400 different things on it, and at least 30 of them have TF antigen, right? And so it didn't bind anything else except for that. So we decided to make a monoclonal antibody. So we're now in the process of making a monoclonal out of that particular construct. So we're hoping to get something that could be a decent reagent to be able to at least image or stain pancreatic tumors. So the design of these particles are like such. If you just put the TACAs on there, just put the tumor-associated carbohydrate, you don't get an immune response in animals. If you just put glycopeptides, which I think is a better antigen, you still don't get it. But if you actually design it properly, put the right adjuvant on it, you actually get a response. And so we're in the process of making second-generation particles to enhance this response. And again, this monoclonal is being done. So let me just finish with the last couple of slides showing this this inhibition of metastasis that I mentioned earlier on. So we actually did this in a 4T1 breast cancer model. It's a great model, and we can discuss that later. And it turned out we got this really nice response where treated lungs looked really good, untreated looked lousy. Then we did it again. It wasn't statistically significant. We did it again. We still didn't find it. We said, okay, we want to redesign these particles. So we redesigned these particles. We put them on the amino acid like I showed you before, and we also made these hybrid particles with Shalvik Biswas. Our controls, like I showed you before, like I mentioned before, is just the particle with linker on it. So it just exposes OHs but no sugar, okay? We've taken these and treated them in different cell types that have either plus or minus galectin-3, that the protein I told you about before. They're more cytotoxic to galectin-3 positive cells, and it turns out the one that we attach to threonine, where R is equal to CH3, is much better than serine, and that's something that we can also discuss. So, and the killing is through apoptosis, but only in the threonine case, as shown here. So in other words, serine and threonine, you don't think that's a big difference. It's only one little method, but it makes a big difference. And I'll show you where it makes a difference here. We actually did an animal study with these, and now we use 4T1 that has Luke2, which is transfected with firefly luciferase, so we can do just fluorescence. And it turns out that if you look at serine, threonine, this is just the linker, and this is PBS. Mice survive after 5 weeks much better with threonine than serine. Really amazing. So we're actually repeating this experiment now to prove that that's correct. So we really like that. And so what's up with this? I mean, I just finished to tell you that it turns out there's a handful of reports that suggest that the structure is very, very different if you have a carbohydrate alpha linked to a serine and a threonine, okay? And we actually proved this many years ago, it turns out. Almost 20 years ago, we published a paper which showed that, and many other structural papers that do NMR on glycopeptides have shown that. We have another very interesting molecule that I'll talk to you about after the meeting, if you'd like, tonight, which is called APF. This is also a peptide that's found in a particular disease state that has the TF antigen on it. We've done a lot of studies with this, and it turns out that what happens is this particular sugar is on a threonine, so there's a methyl group here, and if you actually take that and you consider that 100%, if we move that to serine, we drop the activity by 10,000-fold, okay? And we figured out that, with Alex McCarroll, when we did some computational studies in some NMR, we found out that the sugar is in a very, very distinct conformation when it's on threonine. And when you make it on the serine, it turns out it loses that conformational restriction, so it's much more flexible. So there's a lot of issues with serine and threonine. It's something that I think is very important in biology. So we can talk about that, like I said, at the break, but we don't see this in the serine derivative. So I'm probably a little bit late, so let me just conclude by saying that TF antigen is a valid anti-cancer target for both immune-based and anti-adhesive approaches, and we can use nanoparticles, different types. Gold might not be the best one, but there's several different particles one can use to do this kind of work. The development of new TF antigen-based vaccine constructs, I think, is promising because I know this will work. It's just I think we need the right construct to have a particular to get the best efficacy, should I say? And comparison of serine and threonine is like really, really interesting in O-linked amino acids. This is only an alpha-linked O-linked amino glycopeptides. Suggests a distinct difference in conformational preferences that translate to biological activity. So with that, I'd like to again thank the organizers, thank everyone for coming, and I'll be happy to take some questions. Thank you.
An ignorant question, the TF antigen, is it produced by the tumors of any tissue, or is that tissue or tumor specificity?
Yeah, it's a good question. It turns out that it's on many, many different solid tumors, but it's not, you don't see it in every tumor, right? Because it depends on, as I mentioned briefly in the beginning, it depends on what aberrations or what mutations you have in transferases or in chaperones that actually help those transferases move the sugar on, right?
But what about brain against liver tumors, for example? You have it in both of them in general?
Brain against liver. That's a good question. I don't know.
And usually in glycopeptide and glycoprotein biosynthesis, you have cell type specificity.
Sure.
Therefore, my question.
Yeah, no, I understand. And I know in myeloma multiforme, I don't think there's much expression there. In liver, I know there's expression. But again, it's also tumor type specific too. Yeah, so
tissue type specificity, you have to tissue type specificity.
yes, I mean, yeah.
A very interesting talk, and it's excellent. I have a few questions with regard to, you mentioned about metastasis. And have you looked in your animal models in terms of tumor neovascularization? Do you see any of the blood vessels sort of being targeted with this kind of treatment?
It's kind of interesting you mentioned that, because I just had a meeting with the small animal imaging program where we're starting this other study, where what they want to do is they want to do more pharmacokinetics and they want to actually image this as it goes along. You know?
Right.
Because gold is kind of one of those funny, it's a funny beast because you can't put a fluorescent tag on a gold particle because it quenches the fluorescence.
Right.
If you lose it, then you can get fluorescence. So that's a way of actually seeing if you have to lose things from the gold. The only way you can do it is you can do histology. You can look at like little black dots, right? I mean, you can see those. So we have not looked at neovascularization. This is a very, very well-known model for T1, right? You probably know it. And one thing that is known about it is that it's one of those tumors that expresses TF. It responds very well to antibodies to TF. So antibodies to TF from our other collaborator actually, she showed in vivo that they're highly anti-metastatic and they have like a therapeutic. As a matter of fact, started companies to use that antibody against this. But what we haven't looked at the actual vascular.
I have another question with regard to TACA-coated gold particles. A lot of the tumors develop this EMT, epithelial mesenchymal transition. And in that transition, especially the mesenchymal, do you think these sugar structures are different from? And so, do this particular therapy targets the mesenchymal type?
No one has ever actually specifically tried to do that. One thing that's very interesting too is cancer stem cells. It turns out people have looked at all the stem cells now and looked at the expression of these, and it turns out that it's again specific for the particular tumor. But you do get these on stem cells also. So people are trying to target those also to stop them from proliferating.
Question here.
Well, as you mentioned, TF is just one of the tumor-associated carbohydrate antigen. And can you comment on what has been done in the TN versus, and also TN immune strategy?
TN has been much more well studied, right? And sialTN, as a matter of fact, there was a company in Canada called Monero which had a therapy for an anti-tumor therapy based on sialTN. That was, as a matter of fact, I think that company went away. Some of you might be able to tell me more. But that also didn't make it when it comes to getting toward the clinic. There are ad nauseum synthetic different glycopeptides with TN and sialTN. And also, some of the things that's being done now is that TN is being put on zwitterionic polysaccharides. You know about the sort of castrated carbohydrate. It showed that it turns out carbohydrates have always been known as T-cell independent antigens. But it turns out they can actually be displayed on MHC, I believe. And so put them on the zwitterionic platform, as a colleague of mine, Vito's been doing. He's actually getting very good antibody responses actually to those. This is a TN. He's also doing TF. But yeah, when it comes to your question is when it comes to therapeutics, something that actually is viable, there's nothing yet that is much more done with TN. I mean, TN has been put on virussite particles, on virusomes, all different things.
Because based on my understanding, TN is a one that's probably most likely in the tumor. And because normal cell, you have some TF. And so that's what I'm thinking.
You know what TF is? TF is in your gut. So the microbiome now comes into play, right? So TF is on lots of bacteria in the gut. And it turns out that there's a German group that is trying to develop a vaccine based on gut bacteria because they feel that the display, the presentation of TF on that bacteria is much more akin to what you'll see on other cell types. So if you raise immune responses, they should be more efficacious. I like that idea a lot. So, we'll see what comes of that. But there's other like local wage and many other larger ones too that are fairly important that people have done lots of. Any other questions?
I want to thank everyone.